Bismillahirrahmanirrahim. Dear students of 9C, Assalamu alaikum. I am your teacher Muhammad Ilyas, and this is our sixth interaction regarding biology for Cambridge. So, dear students, today's topic is transport, and we will focus on the practical work. And this topic is taken from your book Practical Biology, a course for O level, chapter number 7, and the topic number 7 according to CI syllabus. Dear students, we will be able to inshallah understand the practical procedure, the apparatus requirement, and to solve the questions given in the practical book. We will focus on the circulatory system as a system of tubes, pumps in the walls which ensure the one-way flow of the blood. Secondly, we will inshallah focus on the composition of the blood, specifically red blood cells and the white blood cells, and observations will be made under the light microscope. Thirdly, we will inshallah make our observations under the light microscope, and we will inshallah try to understand the flow of the blood in the fins of the fish or tadpole, how the blood flows in the capillaries, and inshallah we will observe under the light microscope. So these are our objectives for today's session. So we are going to review the human circulatory system, a system of tubes, pumps in the walls. As you know, the tubes means the vessels. Dear students, there are three main types of blood vessels. First one is the arteries, which carries the blood away from the heart and take it to all other parts of the body. Second, the veins. They carry the blood away from all other organs of the body and return it to the heart. And thirdly, the capillaries. Capillaries carry blood through all the organs of the body, linking arteries and the veins. And their main function is to provide the space for the exchange of materials between the tissues and the blood. The largest artery in the body is aorta, which carries oxygenated blood from the left side of the heart, and it divides to produce smaller arteries and supplying the blood and nutrients to all part of the body organs. And secondly, the largest vein in the body is the vena cava. In fact, there are two of these veins, a spirovena cava, returning deoxygenated blood to the right side of the heart from the head and neck region, and inferior vena cava, which returns deoxygenated blood from the rest of the body. Dear students, now we are going to focus on the heart. The mammalian heart is a muscular pump, and it is made of a special type of muscles known as cardiac muscles. The heart pumps blood around the body so that the blood can supply the tissues with oxygen, nutrients, and other substances. And it also removes waste materials such as carbon dioxide. The bulk of the heart muscles is made up of two lower chambers of the heart, and these chambers are called ventricles, which pump the blood out of the heart through arteries. There are two upper chambers called atria, which are smaller and less muscular. These chambers receive the blood through veins. On the surface of the ventricles are small blood vessels called coronary arteries. These small arteries come off the aorta and supply the heart muscles with the blood. The left and the right side of the heart is separated by a muscular wall called the septum. Between the atria and ventricles are atrioventricular walls. On the right side, this wall is called the tricuspid wall because it is having three flaps, while on the left side of the heart, this wall is called mitral wall or bicuspid wall because it has two flaps. So this is the little discussion about the structure of the heart and its walls. Now I am going to tell you the function of the walls. Walls carries the blood one way. because walls don't allow the blood to move back towards the atria. When the blood comes out of the atria and enter into the ventricles, these valves helps to block the flow of the blood back into the atria. So this is very important function which these valves performs. Now we are going to focus on our practical work, the investigation 7.1 according to your book, and this is to examine the mammalian blood. We will inshallah examine the blood, and the apparatus which we required is a prepared slide of the mammalian blood smear and the light microscope. The procedure will be as follows. Number one, we will examine the blood smear under a low power microscope and then under high power. Note the shape and size of the cells that occur in large numbers, and these are the red blood cells. And dear students, you have to make a label drawing of the red blood cell. So after following this procedure, you will observe the micrograph which I selected for you, the red blood cells and the white blood cells under low magnification. You can see these are the circular red dots. These are the red blood cells, and they are small. These are very small in size, and you can see some large dots which are more darker than the red blood cells, and these are the white blood cells. If we look into the through the high magnification microscope, the red blood cells you can see and the white blood cells. Now the picture is showing you the more clear shape and structure of the red blood cells and the white blood cells. This is the blood smear of human under light microscope. Now we are going to focus on the shape and size of the red blood cells. The red blood cells are also called the erythrocytes. According to this slide, you can see the red blood cells are flattened by concave discs, lack nuclei and mitochondria, and the diameter is 7 to 8 micrometer. These cells are very flexible so that the red blood cells can move through the smallest capillaries in the body. The lifespan of the red blood cells is 120 days. And these are present 4.7 to 5.5, and some books 5.2 it is mentioned millions per cubic millimeter of blood. So this is the description about the red blood cells. Now we are focusing on the functions of the red blood cells. The main and the major function of the red blood cells is to transport oxygen to cells and tissues from lungs, and the carbon dioxide from cells and tissues to the lungs. Beside this, they are smallest in size, as we have discussed earlier, and the red blood cells constitute 99% of the blood cells. Dear students, now we are focusing on the white blood cells under the light microscope. Scattered among the red blood cells are a small number of white blood cells, each with a nucleus. Look for three different types of white blood cells. They can be recognized by the shape and size of their nuclei. Make a label diagram of the three types of white blood cells. So these are the some little description about the white blood cells, how they look like under the microscope. This is the, you can see the white blood cells along with the red blood cells. Different types of white blood cells are present in the blood smear of mammals: eosinophils, neutrophils, basophils, lymphocytes and monocytes. This is the diagrammatic representation of these cells so that you can understand the different types of white blood cells found in the blood of mammals. Now you can see this nucleus present in the cells. Monocytes, they have kidney shaped nucleus. Lymphocytes, they have very big nucleus in the center, and both of these are having cleared cytoplasm. While the xenophils, basophils and neutrophils, their cytoplasm is granular. Granules are present in the cytoplasm and their nuclei is lobed. Dear students, I have selected one more slide for the functions of the white blood cells along with the type of the nucleus they are having and along with the diagram. You can see neutrophils, they have multi-lobe nuclei, and their function is to control the bacterial and fungal infections. These are the most common first responder to the microbial infections. You can see their diagram as well. Eosinophils have bilobe nuclei. Parasitic infections are controlled by eosinophils and allergic reactions. Basophils may be having bi-trilobed nuclei, and their function is to control the allergic reactions. They control the release of histamine, causing the vasodilation. Lymphocytes, deep staining eccentric nuclei, and operate primarily in the lymphatic system. Monocytes, they are having kidney-shaped nuclei, and the main function is the phagocytosis of the pathogens, presentation of antigens to T cells. Eventually they become tissue macrophages, which remove dead cells, debris, and attack microorganisms. Here you can observe the shapes of the white blood cells along with the shape of their nuclei, and you can focus on the functions performed by these white blood cells. Now we are focusing with cells. This slide will show you the differences between these two different types of blood cells. The source of red blood cells and the white blood cells is same. They are made in the bone marrow from the stem cells. While we talk about the different shapes, according to the shape, WBCs are irregular with lots of extensions. RBCs are all biconcave discs. WBCs has nucleus and RBCs has no nucleus. WBCs has no hemoglobin while RBCs have hemoglobin. So these are the differences, a few differences between WBCs and RBCs.